All right, welcome back, everybody. Before break, we have stated that we've got 4 different types of connective tissue. Remember, those are connective tissue proper, cartilage, bone, and blood. Connective tissue proper is going to be subclassified into 2 main subcategories. Those are the you see on here, this is the classification. Proper is going to be classified as loose connective tissue or dense connective tissue. And this is going to be according to the proximity of the fibers to one another. What I mean by this, how close the connective tissue fibers are going to be located next to each other. So for example, this one, if I told you that loose connective tissue, I have large spaces between the fibers. Large spaces between the fibers. Would you consider this tissue that you see here on the right as a loose connective tissue or as a dense connective tissue? Do you have spaces between the fibers? Loose, right? How about this one? Again, we're not looking at how close everything is related to one another. We're looking at how close are the fibers are located to one another. So here, the fibers, do they have spaces in between, or they are closely packed against one another? They have lots of space. So this is going to be considered as a type of loose connective tissue. How about this one? Do I have a lot of space between the fibers, or the fibers are closely packed against one another? Closely packed. So this is going to be considered as a type of dense connective tissue. How about this one? Do you have large spaces between the fibers, or the fibers are closely packed against one another? Closely packed. So this here is going to be dense connective tissue. So this is going to be a dense connective tissue because you see on here no much spaces between the fibers. Here is the same concept. No much spaces between the fibers. How about in here? You have large spaces between those fibers, or you don't? No. No much spaces between the fibers. So this is going to be considered as dense connective tissue. So you see here, according to what? According to what would I be classifying the proper or connective tissue proper? Is going to be according to how close fibers are to one another. Let me show you a confusing one on here. How about this one? What do you think? Loose, dense? What do you think about this one? And loose and dense is going to be determined according to what? According to proximity of fibers. Do you see fibers in here? Do I see fibers? Is there any fibers that you can see on this slide? I can't see fibers. I see cells, large cells. Yes, the cells are closely packed against one another, but I can't see the fibers. So what actually I see is something like this. Fibers and a lot of cells. Even those cells are compressing the fibers. Large numbers of cells, large vacuolated cells. And this is what I'm looking at. Large vacuolated cells. I don't even see the fibers. So this means what? This means that the fibers are located close to one another or away from one another. If the most of the space is occupied by cells, so this means that the fibers are located away from one another, so they have a lot of space in between. So this is going to be considered as a loose or a dense connective tissue. What do you think? If I do have a lot of space in between occupied by cells, would this be considered as loose or dense connective tissue? Remember, if the fibers did have a lot of space in between, so this is going to be loose. If this fibers had no space in between, this is going to be considered dense. So here I'm telling you that all the space is occupied by cells, not fibers. And I'm looking here at those cells. So this means that the fibers are located away from one another. Away from one another. So the fibers here located away from one another. So this type of tissue is going to be a loose connective tissue. Why? Because all this, which has been occupied by cells, is the space between the fibers. So the fibers are located away from one another. So it's a loose connective tissue. Does this make sense? All right. So loose connective tissue and dense connective tissue. So what are the different types of loose connective tissue and dense connective tissue? First type of loose connective tissue, I have collagen fibers and elastic fibers. And you are now able to identify elastic and collagen fibers. You see, those are collagen fibers. The thin, darkly stained long fibers here are elastic fibers. So if I see collagen and elastic fibers, and I have lots of spaces between those fibers, this is a type of loose connective tissues that we call areolar connective tissue. Scattered between those fibers, I can see cells. And those cells are actually the cells that are producing the fibers. So we call those cells, those are the ones producing the fibers, fibroblasts. So if I see collagen fibers, elastic fibers, a lot of cells in between scattered, and I have large spaces between those fibers, this is what type of connective tissue? This is an areolar connective tissue. All right, so how about this one? Do I first see a space lined with cells? You see a space lined with cells. Can I consider this as epithelium? What do you think? Can you consider this as epithelium? Do you have a space lined with cells? Actually, one of the most common wrong answers that I see here is simple squamous epithelium. So some of you think that this is a space and it's lined with cells. Some of you will think that this is a space and lined with cells, but if you notice on here, let's get closer to here. I don't see cells lining the space. What I see in here is a space, yes, but it's inside one cell. And what you see on the side in here is the nucleus of the cell. This is a fat cell. A fat cell is filled with triglyceride, stored form of fat that occupies most of the size of the cell, and that will be compressing the nucleus in the peripheral part of the cell. So what am I looking at in here? It's not a space lined with cells. I'm looking at a space inside one cell. I can see only one nucleus. It's not many cells. Is there a tissue formed by one cell, just one cell? No. You have to have many cells of the same type. So what we're looking at in here, it's just one cell. And you see, I didn't see much fibers. This space is occupied mainly with cells. So we considered it as a loose connective tissue. I don't have much fibers. So if you see a loose connective tissue and the space is mainly occupied with fat cells, and how would I know that those are fat cells? Again, those are large vacuolated cells that have the nucleus compressed against the wall of the cell. So those are my fat cells. You see fat cells, this gonna be the adipose connective tissue. How about in here? I see larger spaces, so this is a type of loose connective tissue. What is the type of fibers? If you remember in here, what is the type of fibers that you can see? What kind of fibers are those ones? Short, branching, and they are darkly stained, thick fibers. Those are my reticular fibers. So if I see reticular fibers, this tells me I am looking at a type of connective tissue that we call a reticular connective tissue. All right, and this is one of the most common wrong answers. A lot of you on the exam will remember the type of fibers. So when I ask about the type of fiber, you answer reticular fibers. But when I ask about the type of tissue, many of you do not remember reticular connective tissue. So remember, if you have reticular fibers in a loose connective tissue, you have spaces between those reticular fibers, the connective tissue is going to be a reticular connective tissue, the same name like the fibers. So reticular fibers are present in, again, what kind of connective tissue? Reticular connective tissue. All right, so again, on the exam, I will ask you, identify this fiber. So what's going to be your answer? Reticular fiber. I'll ask you, identify this type of tissue. What's going to be your answer? It's a reticular connective tissue. Easy, easy. All right, so moving on to the types of connective tissue in which I don't have much spaces in between the fibers. Fibers are closely packed against one another. So this is a dense connective tissue. What do you think? Are those collagen fibers, elastic fibers, or they are reticular fibers? As we mentioned, again, those are collagen fibers. So I have collagen fibers. This is what I first see. I did check whether do I have spaces between the fibers or not. I figured out that I don't have spaces between the fibers. I check now what is the type of fibers. I've seen that those are collagen fibers. If I have collagen fibers and those fibers are closely packed against one another, I need to step back and think here. Do I have a regular pattern of arrangement of those fibers? Is there any pattern I'm following? Yes or no? What do you think? Yes. How about in here? The dense, I also have collagen fibers. You see the diameter is much greater than the diameter of a nearby cell, so it's a thick fiber. So those are collagen fibers. And remember, here, when I have collagen fibers in a dense connective tissue, what I need to do, step back and look, do I have a pattern of arrangement here of those fibers? What do you think? Is there any pattern of arrangement similar to something like this? Is this a regular or an irregular arrangement of the fibers? Irregular, right? Irregular. So we will classify here dense that has collagen fibers according to whether there is a regular pattern of arrangement of those fibers or they don't follow any regular pattern, so they are irregular. So we classify the dense connective tissue that has collagen fibers into two categories, either regular or irregular. So we're gonna have dense regular connective tissue or dense irregular connective tissue. How about this one? I've seen that I don't have much spaces between the fibers. I look closer at the type of fibers. I've seen that those are elastic fibers. So the type of dense connective tissue that is formed of elastic fibers we call this is my elastic connective tissue. So again, what are the 6 different types of connective tissue proper? We have areolar connective tissue. We have adipose connective tissue. We've got reticular connective tissue, dense regular, dense irregular, and elastic connective tissue. Those are the 6 different types of the connective tissue proper. Any problem on how to identify each one of those? So again, first thing I need to look at, is it epithelium or not? Do I have a space lined with cells? I don't see a space lined with cells. So this is not epithelium. I see fibers. So what type of tissue has fibers? Connective tissue. So I need to think here. Is this cartilage, bone, or blood? And we're going to see how to know them in a second. We're going to know that those are not going to be cartilage, bone, or blood. The other ones have very characteristic appearance. So it's going to be a proper connective tissue. It's not cartilage, it's not bone, it's not blood, it's not epithelium. So what would I think of? I think of how close are the fibers to one another. Are those closely packed, or they do have space in between? They do have space, so it's going to be a loose connective tissue. But we have three types of loose connective tissue. I need to memorize this. So when I look at a loose connective tissue, I'm picking from 3. So the list has been reduced from 6 to 3, just by looking at how close the fibers are to one another. Have 3, either areolar, adipose, or reticular. So first one did have collagen fibers and elastic fibers. Do I have collagen fibers and elastic fibers in here? Yes. So this is going to be areolar connective tissue. About this one. About this one. I don't see much fibers, but the whole thing is occupied with large vacuolated cells. Still not epithelium, still not dense. So this is going to be the one that has large vacuolated cells, fat cells. This is going to be my adipose connective tissue. About this one. Large spaces between the fibers. This is going to be a loose connective tissue. What kind of fibers do I have in here? Those branching fibers, reticular fibers. So remember, what type of connective tissue has reticular fibers? It's my reticular connective tissue. How about in here? Bundles, collagen fibers. They are closely packed. They are closely packed to one another, so it don't have much spaces in the connective tissue. So this is going to be dense, not loose. And remember, if I have collagen fibers, I need to step back and look at, is there any pattern of arrangement? Are those dense collagen fibers follow a pattern in their arrangement in the tissue? Yes, if your answer is yes, so this is going to be dense regular connective tissue. About this one, don't have much spaces, still dense. Look, what kind of fibers? Thick fibers. So those are collagen fibers. So collagen fibers, I step back, look at the whole slide. Is there any pattern of arrangement here? No. So this means I'm looking at dense irregular connective tissue. About this one, I'm looking at no spaces between the fibers. No spaces between the fibers, right? So it's going to be dense. Next thing I need to look at is whether or not it's a collagen. What kind of fibers do I have in here? Inside the tissue, those are thin, darkly stained long fibers. So those are elastic fibers. Looking at elastic fibers. So elastic fibers were located in which type of dense connective tissue? They were located in my elastic connective tissue. All right. This is a way by which you would be able to figure out what kind of tissue you're looking at. All right. You can say dense elastic, you can just say elastic connective tissue, both are fine because we don't have another type of elastic other than dense. The same concept with reticular. You don't have to say loose reticular or loose areolar or loose adipose. So you can simply say areolar connective tissue, adipose connective tissue, or reticular connective tissue because once you say those names, I will figure out immediately that it's a loose connective tissue. It's a type of loose. So what needs to be differentiated is regular and irregular. So this is the only time when we're going to be using, we must use dense, because those are two different types of dense regular and dense irregular. All right, and both of them are going to have collagen fibers inside. All right. Questions? All right, so looking at the cartilage, how would I identify the cartilage? So very characteristic appearance of the cells. You see a cell, and this cell is surrounded by space. Cell surrounded by space. What do we call the cells of the cartilage? Remember, something related to the cartilage is chondro, and a cell is site. So this is a cell of the cartilage. And those cells of the cartilage are always going to be surrounded by space. Called the space is the lacuna. So we are looking here at the chondrocytes in lacuna or lacuna. This tells me that I'm looking at the cartilage. So if you see here a chondrocyte surrounded by space, a cavity, this is going to be a chondrocytes in lacuna, and you are looking at the cartilage. So can you see here chondrocytes in lacuna? Chondrocyte here, and surrounded by space. Chondrocyte and surrounded by space. So what I see on here is a chondrocyte in lacuna. So this is a cartilage. This is what I know about this one. Cell surrounded by space. Cells surrounded by space. Space, and inside we've got the cell on here. So what do we call the cells in the space in here? Those are my chondrocytes in lacunae. So if I have chondrocyte in a lacunae, This tells me I'm looking at what again? At a cartilage. Did I have chondrocytes and lacunae here? No. Do you have it in here? Cells surrounded by space? No. How about here? Those are cells squeezed between the fibers. How about in here? Those are cells scattered between the fibers. They are not surrounded by the lacunae, this rounded space. Not in here, not in here. So I can't get the cartilage confused with any of the other connective tissue properties. You see on here, it's very clear. Cells and space. Cell surrounded by a lacuna. Chondrocytes in lacunae. Chondrocytes and lacunae. This tells me again, I'm looking at a cartilage. We have 3 types of cartilage tissue in the body. When you identify that this is a cartilage, what is you should be doing next? You should be looking at the surrounding matrix on here. The surrounding matrix. Do you see any fibers running here in the background? Or does the matrix has a homogeneous consistency? It's almost the same color. I don't have darkening of fibers traveling in between the cells. What do you think? Do you see fibers between the cells in the matrix? I don't see much fibers actually. So the matrix is homogeneous. It has all the same color, same consistency. So homogeneous with H, homogeneous matrix. You don't see fibers. You do have fibers, but you can't see them. So prominent. If I don't see fibers, this is gonna be my hyaline cartilage. Hyaline cartilage. About this one. Do you see chondrocytes in lacunae? What do you think? Do you see chondrocytes in lacunae? Do you see a cartilage cell, a cell surrounded by space? Yes. So this is a cartilage. What is the next thing after I identify that I'm looking at a cartilage? I look at the surrounding matrix. Look at the surrounding matrix. Does it have the same consistency? Is it homogeneous? Or do you see fibers traveling in the matrix? What do you think? Do you see fibers? Yes or no? Yes. So you see fibers. What kind of fibers were those ones? Thin, darkly stained long fibers. Those are my elastic fibers. Elastic fibers. So I have a cartilage and I have elastic fibers in this cartilage. So what would we call this cartilage? Cartilage that has elastic fibers. I have elastic fibers. So we're gonna call this cartilage is my elastic cartilage. No fibers, homogeneous consistency of the background, it's hyaline cartilage. elastic fibers, those thin, darkly stayed long fibers running in the matrix. This tells me I'm looking at elastic cartilage. Elastic cartilage. How about this one? About this one? I see chondrocytes in Lacunae. I see chondrocytes in spaces. I see chondrocytes in spaces. So this is a cartilage. How about the fibers in the background? Look at the matrix. What kind of fibers do you have in the background? Those thick bundles of fibers, those are exactly collagen fibers. Actually, those are not only collagen fibers, they are extra collagen fibers. Extra collagen fibers, because hyaline cartilage has collagen fibers in it, but this is an extra amount of collagen fibers. That's what will make it so prominent. So extra collagen fibers in a cartilage. This is a cartilage with fibers, cartilage with extra fibers. So we're gonna call it fibrocartilage. So again, what am I looking at in here? We're looking at the 3 different types of cartilage. How would I know that I am looking at the cartilage first? I have chondrocytes in Lacunae, cells surrounded by space, and this is very characteristic for cartilage. I look at the matrix. If I had homogeneous consistency, the same color all over, this is gonna be hyaline cartilage. I don't see prominent fibers. How about here? Chondrocytes in Lacunae. Elastic fibers are present here between the chondrocytes. This is gonna be a cartilage with elastic fibers. It's my elastic cartilage. How about in here? Chondrocytes in Lacunae, yes. Thick bundles of fibers surrounding those cells. So this tells me I'm looking at cartilage that has extra fibers, collagen fibers, because this is my fibrocartilage. So again, what are the 3 types of cartilage that we're looking at? We have hyaline cartilage, elastic cartilage, and fibrocartilage. Last 2 types of connective tissues that we're looking at today, those gonna be the bone and blood. Bone or osseous tissue, what does this look like in your daily life? What does this appearance looks like? Like a cut section in a tree, right? Cut section in a tree. It has very, very characteristic appearance. You see, it has a central canal. And surrounding each of the central canal, we have the calcified proteins. Those are forming layers like this. Those are my lamella. So plural noun is going to be lamellae. If you're looking at just one layer, it's a lamella. Multiple ones, multiple layers, lamellae. And embedded between those lamellae, you see those dark spots in here. Those are embedded osteocytes. What's an osteo? Osteo means bone and cyte means cell. So if I'm looking at those dark spots in the osseous tissue, those are lacunae, those are spaces, and inside those spaces I have the mature bone cells. Those are called my osteocytes. So again, what am I looking at in here in a bone or osseous tissue? I look at a central canal, and each central canal that I've got is surrounded by layers of calcified protein. Each layer is called my lamella. Embedded in between those lamellae, we've got tiny spaces, and embedded in those tiny spaces, we've got the osteocytes, the mature bone cells. What do we call the whole structure here? The building blocks of the compact bone tissue that we're looking at in here, that is formed by a central canal and lamellae, and embedded between those lamellae is the osteocytes. We call this is my osteon. And what's an osteon? What's an osteon? Those are the building blocks of my compact bone tissue. And again, what do we call this dark circular center in the middle of the osteon? This is called my central canal, and I have got blood vessels traveling through it. What do we call again? The layers of bone. Layers of bone surrounding the central canal. Those are my lamellae. And what do we call the spaces where you keep the mature bone cells, the osteocytes? Those are my lacunae. Where did we hear lacunae before? Do you remember? The lacuna was also surrounding the cartilage cells, the chondrocytes. So looking at the bone under the microscope has a very, very, very characteristic appearance. Have building blocks, osteons, central canals surrounding the central canal. You're gonna have the bone layers. We're gonna discuss the structure here of the compact bone later on in much more details when we get to chapter 6 and 7. All right, any problem with identifying that this is the osseous tissue or bone tissue? I didn't get this, Theodore. What do you mean? No, all right, all right. The middle circle here is the central canal, and the smaller spaces here are the lacunae. And embedded in those lacunae, inside the lacunae, we've got the osteocytes, the mature bone cells. All right. Looking at the blood in here, blood has also a very characteristic appearance under the microscope. It's so hard to get it confused with any other thing. See a clear matrix, and you get blood cells scattered all over place. And what we see on here, we see cells that have dark periphery and pale center. Those cells are my red blood cells or erythrocytes or RBCs. Red blood cells, another name for it is going to be erythrocytes. Erythro means red and cyte means cell. So red cells, erythrocytes. What do I see in here? Cells don't have a nucleus. So the red blood cells do not have a nucleus. They look like a donut here. They have a dark periphery and pale center. Why do I have this appearance as erythrocyte? Erythrocytes are biconcave discs like this. So the middle part of the cell is much thinner than the peripheral part. And remember, inside, what do we keep inside the red blood cells? We keep a red pigment responsible to carry the oxygen called the hemoglobin. And remember, hemoglobin is going to be present in larger amounts in the peripheral parts. If I'm looking here, this is my eye, and I'm looking down at the cell. So those gonna be the peripheral parts of the cell. So they will appear darker in color, as you see on here. And the central part of the cell, this part on here, is much thinner. So it's gonna appear lighter in color. It has a pale center. How about the other cells that we see on here that have nuclei? So it has a nucleus, this one has a nucleus, this one has a nucleus. Those are white blood cells. White blood cells. Another name that we use for white blood cells is going to be leukocytes. Leuko means white. Cyte means cell. Leukocytes are my white blood cells. And we have different types of white blood cells. Unlike the red blood cells, you have only one type of red blood cells, which is a mature form. Normally, we have premature forms that might exist in some conditions, but generally speaking, I have one type of red blood cells. I have many different types of white blood cells. We can see here two of the most common types, lymphocytes and neutrophils. How would I differentiate between lymphocytes and neutrophils? Look at the nucleus. If the nucleus is dark, large, and occupying most of the size of the cell, it's a lymphocyte. If the nucleus is segmented, so it has multiple segments, and doesn't have any specific shape, and the cytoplasm has granules. You see, it's granulated. This is a very simple way to identify the neutrophils. So you need only for now to differentiate between those two types of leukocytes or white blood cells. And if I have segmented nucleus, granules in the cytoplasm, those are going to be my neutrophil. If I have a dark, large nucleus occupying most of the size of the cell, this is a highly active cell. We call this is my lymphocyte. Both lymphocytes and neutrophils are white blood cells. Both of them are different types of white blood cells. And we have many other types of white blood cells that we don't see on this slide. All right, any questions? Any questions? All right, we can have a break for for 10, and we can come back, discuss the location and function of the different connective tissues that we've learned how to identify. All right, so we can have a break for 10, and we're gonna be back to discuss. Yes, we have only 9 questions so far. All right, we will see you in 10. All right, welcome back, everybody. So it seems that we only had 9 questions so far, not 8 questions so far, not 9. So the last question, it was number 10. It was the collagen fibers. All right, did everybody get this? So we have only 8 questions so far, and the last one was the collagen fibers. All right, so let's have a look on here at the different types of connective tissue, their locations, and their function. So first one is that we've seen did have collagen fibers, elastic fibers, fibroblasts in between, and lots of spaces. This is my areolar connective tissue. And if you remember, the areolar connective tissue here has a lot of spaces in between its fibers. And this would serve us well to support the epithelium. If you remember, epithelium was avascular. So in any epithelial tissues that we've seen, I don't have blood vessels. I rely on the support of the underlying connective tissue, and the underlying connective tissue of choice in here, it's gonna be the areolar connective tissue. Why? Because simply I have a lot of spaces between the fibers, which will allow the fluid carrying the oxygen, carrying the nutrients to diffuse up towards my epithelium. All right, so I won't have something like this, for example. Yes, there is no much space for the fluid to travel up to my epithelium. So here, the areolar connective tissue is going to be widely distributed under the epithelium of the body, which is going to be covering all the organs and lining all the internal surfaces. Moving on to the adipose connective tissue. Remember, I don't see much fibers. The tissue is mainly occupied with fat cells. And again, fat cells on here are large vacuolated cells, which nuclei are compressed against the wall of the cell. What's inside the adipose tissue cells? It's triglyceride, it's stored fat. What is the function of the adipose tissue? The function here will be to reserve energy in the form of fat. So if I have excess intake of energy, for example, I did eat 2,000 calories a day and my daily need of calories is 1,000 calories. So what you're gonna do with this extra amount of energy intake that you've got is gonna be mainly stored as fat, not only, but mainly stored as fat. Also, the adipose connective tissue supports and help with the protection with different organs. Where is it going to be located? It's widely distributed underneath the skin, under the skin, in a layer called the hypodermis, under hypodermis skin. It's also going to be supporting the internal organs like the kidneys and eyeballs. You're keeping the eyeballs in their orbits, surrounded by fat for them to not move around in this bony socket. Also, you're going to keep the kidney in its place by stabilizing it by fat tissue, by adipose connective tissue, for it to not move around in the abdominal cavity. Also is going to be in the abdomen, surrounding other abdominal organs, and it's going to be located in the breast. And please pay attention on here that it's located in the breast, not the mammary glands. All right, so you see on here the breast structure. It has glands, and we call those glands are the mammary glands. And remember, glands are formed by epithelium. We call this the glandular epithelium. So mammary glands are formed by glandular epithelium, but the surrounding fat that will be holding the mammary glands in the breast, this is going to be formed by adipose connective tissue. Moving on to the next tissues that we've seen branching fibers, and they have a lot of spaces in between. In those spaces, you're going to have a large number of blood cells, mostly going to be immature blood cells. So all those are immature blood cells, mainly going to be white blood cells. Where can I find the reticular connective tissue? It's going to be mainly in my lymphoid organs. Like what lymphoid organs? Like the spleen, lymph nodes, and bone marrow. And those are actually the sites in your body where you keep large reserves of white blood cells. In the bone marrow, for example, this is where I have all the stem cells responsible for the generation of white blood cells, red blood cells, and platelets. All those are going to be produced from the stem cells located in your bone marrow. So reticular connective tissue is located in my lymphoid organs. And actually, the slide here tells me everything. It tells me this is a reticular connective tissue when you look and identify the type of fibers. And as you look around, you will see a lot of immature white blood cells, a lot of cells. Those are immature white blood cells. And where do I have immature white blood cells? In my lymphoid organs, my spleen, lymph nodes, and bone marrow. And this explains why if you've got an infection, you're going to have swollen lymph nodes because the cells are going to be rapidly proliferating, causing the inflammation of the lymph nodes. So they will be swollen, inflamed due to the active replication of the white blood cells. You are forming an army to fight the infections. Looking at our next type of tissue, remember, I've got fibers that don't have spaces in between. We classify this as a dense connective tissue. And remember, we looked at the type of fibers building up this tissue. We have collagen fibers. So what exactly is your question here? Can you repeat, please? I didn't get your question. So you're asking about the previous slide or what? All right, so yes, what's the question? All right, so again, we're looking here at dense connective tissue, which I have collagen fibers, thick bundles, And if we notice on here, we have compressed cells. The cells are squeezed between the fibers. Those are the cells that are responsible for the production of the fibers. So we call them my fibroblasts. What did we name this tissue? If you remember, this is my dense regular connective tissue, as the collagen fibers are following a pattern, a regular pattern of arrangement. So what is the function of the dense regular connective tissue and where is it located? If you go to the gym, when you train your biceps, you're gonna be training it like this or like this. What do you think? Like this or like this? So A or B? So what happens if you decide to train it like this? B. B actually gonna be activating much more fibers in the biceps. So enhancing better enlargement in the size of the biceps. But why would I choose A, not B? Because simply the tendons and ligaments that are attaching here the muscle to the bone and attaching the bones to one another are going to be formed of dense regular connective tissue. And the dense regular connective tissue, it's going to be responsible to withstand tensile strength only if it's pulling in one direction. So what's going to happen if I decided to train my biceps like this? My tendons and ligaments going to be torn. So I cannot tolerate tension in different directions. The tendons and ligaments will be providing me with very good tensile strength when it comes to pulling in one direction. But if you decide to pull in different directions, the tendons and ligaments may rupture. All right, what is the difference between a tendon and a ligament? Simply speaking, a tendon attaches a muscle to a bone. So let's say this is my quadriceps in the thigh. And this is my knee, like this. And this is the leg down in here. This is going to be the tendon of the quadriceps. It attaches a muscle to a bone. This is a tendon. Compared to a ligament attaches two bones together. Like the ACL, PCL in your knee. Anterior cruciate ligament, posterior cruciate ligament. Those are attaching the femur to the tibia. So they are attaching again two bones to one another. So again, what is the type of connective tissue forming my tendons and ligaments? It's a dense, regular connective tissue. dense regular connective tissue. And what's the function here? It stands pulling force in how many directions? How many? One. All right, so looking here closer to this connective tissue, remember, I don't have much spaces. I'm full of collagen fibers. What's the next thing I need to look at? Do I have follow any regular pattern of arrangement of those fibers or not? So here, am I following any kind of regular pattern of arrangement? No. So this is gonna be my dense irregular connective tissue. Dense irregular connective tissue here can withstand tension in many directions. Unlike the dense regular, it withstands tension in only one direction. Like where do I have dense irregular? Like for example, your skin. Your skin was formed of an outer layer. We call it the epidermis. And then we have the dermis. And then below the skin, we had hypodermis. So in the dermis, in order for my skin to not rupture, as I pull on my skin in many directions here, my skin won't rupture. What allows it to do so is the presence of the dense irregular connective tissue in its structure. It can withstand tension in many directions. Another location is going to be the joint capsule. So I can move my joints in many directions without having a rupture in the capsule surrounding the joint cavity. Another is going to be an organ capsule. So when you are running, your organs are located inside the capsule. Why you don't have a rupture of this capsule? Because it can withstand tension in many directions. So my organs are going to be pulled in many directions within their spaces, but as they are surrounded by a capsule of dense irregular connective tissue, this will prevent the rupture of the capsule. So again, where do I have dense regular connective tissue? In my tendons and ligaments. Where do I have dense irregular connective tissue? Where do you need tensile strength and accommodating tension in many directions? In your dermis of the skin, your joint capsule, and your organ capsules. Moving on to another type of connective tissues that we've seen. Seeing dense connective tissue, no much spaces between the fibers. When I look closer at the type of fibers, I see those thin, darkly stained long fibers. So those are my elastic fibers. What kind of tissue has elastic fibers and it's a dense type of connective tissue? It's my elastic connective tissue. Where do you need to have elastic connective tissue and what's going to be its function? The function here of the elastic connective tissue is to allow you to expand and to recoil. Expands, recoils, expands and recoils. So you need something that allows expansion, but also allows you to recoil. Like where? Like in your lungs. Where specifically in my lungs? In the bronchial tubes. In the bronchial tubes, I need to expand, I need to recoil, I need to expand, I need to recoil to regulate the amount of air that travels in. Like what you're saying here, large arteries, exactly. Why large arteries? Did you ask yourself what is meant by the numbers you get when you check your blood pressure? When you check your blood pressure, arterial blood pressure, how many numbers would you get? 1, 2, 3, 5, how many? For the blood pressure reading, you get 2 numbers, right? So let's say 120 over 80 on average. What do I mean by 120 millimeter mercury? And what is 80 millimeter mercury? What does this mean? What do you think? Systolic and diastolic. And what do I mean by systolic and diastolic? So this is called the systolic arterial blood pressure. This is called the diastolic arterial blood pressure. What do I mean by this? Systolic, yeah, both of them are pressures in the arteries, right? Both of them, but what's the difference here? What's by definition systolic? Systolic means high and low. It's not what it means, but it's a fact. Yes, systolic is going to be higher than diastolic. But what does this mean? One rest, one work. But when I take the blood pressure reading, I'm resting, and I still have 2 readings. So systolic means, first, what is arterial blood pressure? It is the pressure of what? Of the blood flowing in the artery. So as it passes in the artery, there is friction between the blood flow and the wall of the artery. So the blood is going to be pushing the artery out. What we're doing, we are surrounding the arm by a cuff. So we're going to be raising the pressure within this cuff to check the force by which the blood is applying on the wall of the artery. So we're surrounding the arm in here to check the pressure of the blood flow that is applied on the wall of the brachial artery. We're checking the blood pressure off. All right. So again, what's blood pressure? What's blood pressure? Blood pressure simply means it is the pressure of the blood flow applied on what? On the artery, on the wall of the artery. So the blood, as it flows in the artery, it pushes the wall of the artery out. This is what we're measuring. The force by which the blood flow pushes the wall of the vessel out. This is what I mean by blood pressure. All right, is this clear? What's blood pressure? All right. What's systolic and what's diastolic? The systolic and diastolic, it tells you when. It answers the question when. So the force by which the blood flow pushes the wall of the artery out during ventricular contraction. So when the ventricle of the heart is contracting, This squeezes the blood out in the artery. So it pushes the wall of the arteries by a pressure of 120 millimeter mercury. This is what I mean by systolic blood pressure equals to 120 millimeter mercury. That the blood flow applies a force on the wall of the artery when during ventricular contractions. This is what I mean by systolic. It's during ventricular contraction when my ventricles of the heart are contracting, squeezing the blood out, of a force equal to 120 millimeter mercury. So if I am applying a pressure of 120 millimeter mercury on the outside, this will overcome the pressure inside, so there will be no expansion and the artery will be partially closed or completely closed. This is the highest pressure that I can possibly get in the artery. So when I apply a pressure over 120 millimeter mercury from outside, what's going to happen? You completely close the vessel. That's why when you are checking the blood pressure, you don't want to keep the cuff inflated, because this simply blocks the blood supply of the forearm and hand. You compress the artery so the blood can't move anymore. The force that the blood is applying to the wall of the artery when it flows during ventricular contraction is 120 millimeter mercury. This is what I mean by systolic blood pressure. What's diastolic blood pressure? Can you tell me? What's blood pressure again? Blood pressure is the pressure applied by the blood flow on the wall of the artery. This is arterial blood pressure. The question now is when, and we're answering diastolic. What I mean by diastolic, it's during, so what was systolic? Systolic was during ventricular contraction. Here diastolic means when the ventricles are relaxing. So this diastole or diastolic means during ventricular relaxation. So you contract and now you are relaxing the ventricles. So the ventricles are relaxing. I measure the blood pressure here. It's equal to 80 millimeter mercury. What this means? That the blood flowing in the artery pushes the wall of the artery by a force of 80 millimeter mercury. This is what I mean by diastolic blood pressure. So again, what's systolic? Ventricular contraction. What's diastolic? Ventricular relaxation. How come do I have blood flowing into my artery when the heart is relaxing, when the ventricles are relaxing? What do you think? If the ventricles are relaxing, am I pumping blood out? Is there any blood that is going to be leaving the ventricle into the artery? What do you think? If I'm increasing the ventricle size, I create actually a suction force. Would I be pushing blood out? Yes or no? If I am increasing the size, I'm not squeezing blood. I'm not squeezing blood. Will there be any blood ejected from me? So if I told you I have a can here, I squeeze the can, so what's inside will come out. If I told you that I will be dilating the can, increasing its size, would there be anything coming out if I'm not squeezing it? No. So there is no blood that gets ejected. So no blood gonna be moving out. How come, my question now, how come there is a blood flow and this flow applies a pressure of 80 if the heart is not ejecting blood? So I know something that you're gonna learn later on, that you have valves that will prevent the backflow. I will prevent the blood from coming back, but I'm not pushing it forward. When I relax the ventricles, how come I have a blood flow and this flow is applying a force of 80 millimeter mercury on the wall of the arteries, although I'm not pumping blood out? This is the question here. So I can understand. I've ejected blood during ventricular contraction, so blood is flowing in the artery. So it will apply a pressure on the wall of the artery. It's going to be equal to 120. I can understand this. But what I can't understand, how come when the ventricles are relaxing, the blood flowing in the arteries will apply a pressure? How come there is, in the first place, blood flowing in the arteries if I'm not pumping blood out? It's a closed system, it's a closed loop, but where is the engine? Is the ventricle. The ventricle is the one that pumps the blood. If you are saying that you have pushed during ventricular contraction and the blood will keep moving by its inertia, is this what you mean? So you push something and it will be kept moving due to its inertia, right? So you push something and you stopped. So if you push, for example, a cart in your shopping center, you pushed it and then you stopped pushing it, it keep going by its inertia, right? Is this what you're saying? Right. The key here is that the wall of the vessel has a lot of resistance, so it will stop. There is a lot of friction. Try to imagine you are pumping this blood and this blood gets distributed everywhere in the body, passing through microscopic vessels. So I have a lot of resistance that I need to overcome. I can't just rely on the inertia. If I am talking about something that is not faced with major resistance, yes, this would be applied, like it can move by inertia. But here, I have a lot of resistance. So if I'm not pumping, if I'm not pushing the blood forward, it will not move because I'm faced with a lot of resistance. So how comes there is a blood flow while you are relaxing your heart? Due to the presence of what kind of connective tissue in the wall of your large arteries? Elastic connective tissue. How? So when I contract, what happens? The arteries, the large arteries will expand like this. Like a balloon. And then when I stop contracting and I start to relax, what's gonna happen to the wall of those arteries that have a dense elastic tissue? Those expanded arteries will recoil back to their normal size. So they will recoil Remember, when I recoil, I'm creating a high pressure. I'm squeezing the blood. And when I squeeze the blood can move backwards and forwards. But here, remember, I have something that blocks the blood from going backwards. I have something called the valves. And when I recoil, this will squeeze the blood to keep it moving forward by a pressure of 80 millimeter mercury. So here, during relaxation, what actually keep pushing the blood is not the heart, it's the large arteries. Large arteries are the ones that will expand during ventricular contraction. They expand, storing energy. For the heart, when it relaxes, they recoil back to their normal size and they push the blood to move forward. So the pump in here, it's not going to be the heart anymore. The pump in here during ventricular relaxation, it's going to be the elasticity of my large arteries. And again, what provides those large arteries with this degree of elasticity? The presence of what kind of connective tissue in their walls? Elastic connective tissue. Does this make sense? Does this make sense? It's like I store some power during ventricular contraction to use it later on during ventricular relaxation. All right, and you see here, definitely the pressure that will be created due to the ventricular contraction is going to definitely be higher than the pressure that will be created due to the recoil force of the large arteries. That's why there is a difference between systolic blood pressure and the diastolic blood pressure. All right, so we're going to stop at this point today, and next class we will start by discussing the location and the function of the different cartilage tissues.